Good day, everyone. I am Julie Christy Lopez, and today I'll be introducing the topic of animal breeding, a practice that plays a vital role in shaping stronger, healthier animal population. It's not just about caring for individuals' animals, it's about improving entire groups over time through science and strategy. Let's start with the main purpose of animal breeding. There are two key goals to remember. First, to improve animal population. This means enhancing the genetic traits across groups, not just a single cow, pig, or chicken. Breeders aim to develop animals that grow faster, resist diseases better, and perform more efficiently as part of the herd or flock. Second, to improve future generations through proper breeding, desirable traits like physical strength, fertility, or calm behavior are passed on. This results in animal populations that are better suited to meet human needs and adapt to changing environments over time. In this part of our report, I'll be discussing the main responsibilities of animal breeders, which revolve around two key tasks: selection and breeding. These are the core actions that help improve not only current animal populations but also their future generations. Let's start with the first responsibility, the selection. Selection is the process of identifying animals that already show desirable traits such as higher milk production, faster growth, better immunity, or calm temperament. Breeders study records, observe performances, and sometimes run tests to choose the best candidates. Through careful selection, breeders increase the presence of good traits in a population. There are different types of selection. The natural selection, where nature filters traits through survival. Artificial selection, where humans guide choices based on their preference. Mass selection, which focuses on an individual's performance. Family selection, based on how relatives perform. And the last one is the progeny testing, which involves evaluating offspring to judge genetic value of their parents. The more accurate and thoughtful the selection, the faster breeders can improve an animal group genetically. And the last one, breeding. Once the right animals are selected, the next step is controlled mating. The goal is to combine strong genetics to produce healthier, more capable offspring. There are several breeding techniques used to achieve this. Pure breeding, mating animals of the same breed to preserve consistency. Crossbreeding, mixing different breeds to enhance traits and boost vitality. Inbreeding, pairing closely related animals to strengthen certain traits, though it must be done with care. Outbreeding, mating unrelated animals to increase genetic diversity and reduce inherited weaknesses.
Hello everyone, I am Jared and I will be explaining what gene frequency is. So, based on description, gene frequency, or also called allelic frequency, is basically how often a certain gene or allele appears in a population. So, it helps us know how common or rare a gene is in a group of animals. Now, here are important things to note regarding gene frequency. First up, the gene frequency value is always between 0 and 1. Anong dili man siya more than 1? Because 1 already represents the whole population. Next, if an allele does not exist in a population, then automatically 0 na gina siya. Next, if it is the only one at its locus, or for example, tanan sa population has that gene, then its frequency is 1. Then, if it makes up 35% of the genes, the frequency is 0.35. So, basically, ato ra siya gi-convert into a decimal. So, now, we have here an example. You have 10 puppies, 4 of which are brown and 6 of them are white. What is the genetic frequency for each color? So, what we will do, atong i-write down ang colors, then i-convert ang values. So, first up, we have our white puppies. We have 6 of them out of 10. That makes up 60% of the population. Then, i-convert na to into a decimal, mimo na siya og 0.6. So, marga pa itong buhaton sa brown. So, now, with our gene frequency formula, ato rang kuhaon ang values, i-substitute, then i-solve na to. So, we have 0.6 plus 0.4 is equal to 1. So, permigi na siya equal to 1. Now, as to why gene frequency is important, breeders can use gene frequency to know if good traits like disease resistance are increasing or decreasing in the animals that they breed. So, that's all. Thank you.
Now, I'll walk you through the part A of our sample problem, which involves computing the frequency of the B allele in a population of blue undulation chickens. We're given a flock of 100 chickens with the following genotypes. 36 are black, 44 are blue, 20 are white. Each chicken carries 2 feather colors genes. So in total, we have 100 chickens times 2 genes each equals 200 alleles in the population. Now, we want to find the frequency of the B allele, which will represent as P. And here is the formula. Let me break this down. From the black chickens, each has 2 B alleles. So we calculate 2 times 36 equals 72 B alleles. From the blue chickens, each has 1 B allele and 1 B allele. So we count 1 times 44 equals 44 B alleles. Adding this together, 72 from black chickens plus 44 from blue chickens equals 116 B alleles. Now, divide that by the total number of alleles. 116 divided by 200 equals 0.58. That means 58% of the alleles in this population are B alleles. You might be wondering, why do we multiply black by 2 but blue only once? Great question. Black chickens carry 2 copies of B allele, both inherited from parents, so we count both alleles. Blue chickens, on the other hand, only have 1 B allele, so we count it once. This approach helps us measure the actual presence of the B allele in the entire population, not just how many animals have it, but how many copies of the gene exist overall.
Good afternoon, everyone. I'm Malika Honyo. I'm going to discuss how to solve gene frequency of white small b allele in blue Andalusian chicken. In this population of 100 blue Andalusian chickens, each chicken has 2 alleles for the reader color gene, meaning there are 200 total alleles. In calculating the frequency of the white allele small b, there are 3 types of chicken based on their genes. 36 black chickens, they carry no small p alleles. 44 blue chickens, each one carries 1 small p allele, contributing 44 small p alleles. 20 white chicken, each one carries 2 small p alleles, contributing 40 small p alleles. So the total number of small p alleles in the population is 44 from blue chicken plus 2 times 20 from white chicken equals 84. We divide the total number of small b alleles by the total number of alleles in the population. 84 divided 200 equals 0.42. This means the gene frequency of the white allele small p is 0.42, or 42% of all the feeder color gene in this flock are the white alleles. Thank you.
Good afternoon, everyone. Good afternoon, Doc. I am Sharil, and now I'll be discussing the genotypic frequency. So, the genotypic frequency is the relative frequency of a particular one locus genotype in a population. Kumbaga, it describes how common a genotype is within the total population. So this helps us in understanding how characteristics are inherited and how common certain combinations of genes are. In genotypic frequency, genotypes are represented by capital letters instead of lowercase letters. So the P represents homozygous dominant genotype, H represents heterozygous genotype, Q represents homozygous recessive genotype. So we're just gonna use these letters lang for easier calculation. So let's apply this using the example ganina, katong a flock of 100 Andalusian chickens. 36 are black with genotype P, 44 are blue with genotype H, and 20 are white with genotype Q. We are going to convert this into frequency by dividing them by 100. For genotype P is 36 divided by 100 equals 0.36. For genotype H, 44 divided by 100 equals 0.44. And for genotype Q is 20 divided by 100 equals 0.20. And then if we add na to sila together, we will get 1.0, which means we have accounted the entire population. And the important rule is that the genotypic frequency should always be equal to 1 since we are accounting for the 100% of the population. And keep in mind Rajud nga different siya from the allelic frequency nga ga-focus ra on individual allele, not the whole genotype. And that ends my discussion on the genotypic frequency.
Population genetics basis. What is population genetics? Population genetics is the study of how the frequencies of alleles, different versions of a gene, and genotypes, the combination of alleles an individual has, in a population change over time. Key terms: gene pool, the total collection of all the genes and their different alleles present in an interbreeding population. Allele frequency, the proportion of specific allele within a population gene pool is calculated as number of copies of the alleles or total number of alleles for that gene in the population. Genotype frequency, the proportion of individuals in a population that have particular genotype is calculated as number of individuals within the genotype or total number of individuals in the population.
That's it for our topic. To summarize it up, the population genetics helps us understand how genes and traits are passed and changed in animal groups. Breeders use gene frequency and genotype frequency to make better breeding decisions. By selecting desirable traits through natural or artificial selection, they can improve animal health and productivity over time. In the long run, if a certain trait becomes very common, it may reach fixation, means one version of the gene remains in the population. This shows selection, breeding, and genetics all work together to shape the future and animal population. And that's all. Thank you.